Hello everyone, I'm Ane Fenter and today I'll be presenting my master's thesis, Do Mixed Species Planted Forests Perform Better than Monocultures During Short-Term Drought Events in Central Panamanian Tropical Lowland Forests? I did this at the Faculty of Environmental and Natural Resources at the University of Freiburg in November 2022, and my second supervisor and second examiner was Dr. Norbert Kunert, Dr. Peter Hajek and Dr. Thomas Seifert. The outline of this presentation is first the introduction of the topic, after which I'll go into the materials and methods and the data analysis. Then I'll talk about the results and what they might mean in the conclusion. Mixed species plots. Studies have concluded that increased species richness in forest stands has a positive correlation with increased productivity and vitality. See papers below, but more insight is needed into species-specific responses to variation in climate. Mixed species plantations lead to increased diameter growth, higher carbon sink storage, better capacity for providing ecosystem services, and higher resistance to drought-related stress, according to the following papers. But questions still remain, like if differences in growth performance exist between stands with 5, 2, or 3 species, what level of mixture is best and for what purpose? Carbon allocation adaptation. The better growth that you see in mixed species stands may be linked to positive diversity effects, i.e. carbon allocation shifts, as a result of environmental conditions and potential competition for limited sources. A key mechanism of forest adaptation to changes in dry periods is the allocation of carbon. Carbon allocation happens due to reduced photosynthetic activities as a result of stress factors. Stresses may either directly or indirectly affect this productivity. Carbon allocation patterns alter in response to environmental factors, leading to the shifts of growth in trees. In the tropics, carbon trade-offs in trees are more influential on season woody biomass production than net primary production allocated to growth, according to Doughty et al. 2014. Tree rings in the tropics. In this thesis, I investigated whether tropical mixed plantations better employed this adaptive mechanism compared to monocultures. Tree rings indicate the concentric addition of biomass to the tree in all the organs, stem, roots, and branches, and can be used to construct growth patterns. Tree ring analysis allows for using past climatic data as reference for explaining patterns in growth as captured in year rings. More importantly, though, it allows for potentially matching weather anomalies with the anomalies in the shape and size of the tree ring in specific years, suggesting the mechanisms by which the tree may have responded to the event. Tree ring formation and their differences from one season to the next can therefore be used to determine seasonal changes in biomass addition to the various tree organs, which you may call carbon allocation. The aim of this research was to determine whether observable variations in year ring increments among different tree organs exist between mixed species plots versus monoculture plots as a response to dry season variability in the tropics, which could allude to the plot's response to dry period variations via carbon allocation. This is towards a better understanding of variability in tree ring widths as a representative of tree response to climate stresses via shifting growth patterns. The hypothesis of the research was, in the first question, comparing the growth patterns among stem, roots, and branches between monocultures and mixed species plots in response to dry season length variation versus a regular year. For this, it was hypothesized that mixed species plots will show stronger indications of carbon allocation changes, which are changes in tree ring increment amongst organs, in response to variation in the dry season lengths from one year to another when compared to the response of monoculture plots undergoing the same variations. Second question was to assess any variability of growth in different tree organs, i.e. stem, roots, and branches, in a monoculture regime in response to variations in dry season length versus a regular year. And the hypothesis was that species avoiding the dry season, which are dry season deciduous species like Cedrela odorata, will show a lower response to dry season anomalies than species that persist the dry season, brevi deciduous, Anacardium excelsum. The study site, the materials and methods. The div tree experiment in the Colón district in Sardinia, central Panama, is one of the oldest experiments of DivTreeNet. In this, they planted 6 indigenous tree species chosen by their relative growth rates, and the trees were planted in diversity treatments in a random distribution. This is specifically designed to test how increases in tree species richness affect the cycling and storage of carbon. And I obtained access to these harvested samples of tree discs from each diversity treatment. This is the map of the area close to Panama City. You can see where the red dot is, that was where the experiment was. The site itself. The area was once a natural forest around 1953, and then it was cultivated agricultural land for 2 years, and then it was converted to pasture land before it became an experimental site. It's 30 kilometers from Barrow, Colorado Island, which has a weather station reporting climatic data for the study site. The average yearly rainfall for this area is estimated at 2,350 millimeters. The dry season, January to March, has a rainfall of 25 to 50 millimeters. Wet season, May to November, a rainfall of 250 millimeters, and in between those two, a rainfall of 120 millimeters. The annual temperature stays consistently around 25 to 31 degrees Celsius. This picture shows the plots and was taken directly from the source of Kunert et al., a 2012 paper on tree diversity that was done on the same DivTree experiment. The grey blocks are the investigated subplots, and on the top left of the picture you can see the different plots and then the species that were planted on them. This plantation was established in 2001 and on a 4.5 hectare area, and this has slow growing species, semi-fast growing species, and fast growing species, each of which are either brevideciduous, semi-deciduous, facultative deciduous, or just deciduous. Each plot was 45 by 45 meters and planted at 1,111 stems per hectare with 3 meters between the trees, which is quite a standard planting. The experiment itself had varying degrees of species mixtures planted in a random distribution, leading to 6 3-species mixture plots, 6 6-species mixture plots, and 12 monoculture plots for each species. But all trees of Cordia aleodora subsequently died after 1 year and were excluded from the study, leading to a final experimental design where I had 3 2-species mixture plots, 3 3-species mixture plots, which were all the T plots, the 4 5-species mixture plots, which were the A plots, and 10 monoculture plots, 2 for each species, leading to a total of 20 plots. The sample collection, just a diagram to show, had 1 disc from the coarse root, if it was larger than 5 centimeters diameter, 1 disc from the stem at DBH, and 2 discs from the branches above the crown. The discs were then taken to the lab, and each was prepared using sanding and then scanned using a Microtech dendrological scanner. The image was then loaded onto Windendro software. Each sample disc was scanned and the year rings were marked from the outermost ring, the last one, towards the pith, the middle. The growth increment in millimeters was measured for each ring. This is a summary of the data that was captured. Data analysis. For 2 hypotheses, I wanted to show any significant differences in growth pattern between organs in the group monocultures across the years 2014 to 2016, in each mixed species treatment, in the A plots, T plots versus the monoculture plots across 2014 to 2016, and species-specific monoculture plots, i.e. only looking at the monocultures of each species across the whole plot's lifespan. The changes in dry season length were identified by increasing number of dry days in each season. A regular year, the control year, was 2014, the dry year was 2015, and the driest 2016. All analyses were performed using RStudio and Microsoft Excel software. The descriptive statistics were first rendered for each treatment, then box plots were created for each organ's growth increment across the 3 years. Any significant changes were identified in quartile spread for each organ growth across the years, and from that I could see which change needed to have a paired sample t-test performed on, and this was at a p-value of 0.05. Firstly, results. Shifts in carbon allocation did not seem to occur in monoculture treatments in response to dry season variations. There were no significant differences found in stem, roots, or branches between the years 2014 to 2016. Secondly, in mixed plots versus monoculture plots, A plots, which had 5 species, showed significant differences in the stem growth between 2014, the regular year, and the drier year, 2015, and significant difference in growth between 2015 and 2016, the drier and the driest year. In the T plots, which had 2 and 3 species mixtures on, there was significant difference in the growth in the stem and the roots between the years 2014 and 2016, which is a regular year and the driest year. Therefore, shifts in carbon allocation may have occurred in mixed treatments in response to dry season variations. For the species-specific monoculture plots, again, no significant differences were detected in any of the species, in any of the organs, across any of the years, and therefore concluded that no shifts in carbon allocation seem to occur in any plot in response to dry season variations. In the growth patterns for each species' monoplot, we determined that all species showed a general downward trend in stem growth in the years 2014 to 2017. The climatic variation in these years were therefore not affecting any one species more than the other. Cedrela odorata, which is dry season deciduous, and Luea siemanii, which is brevi-deciduous, had the same response in growth to the seasonality. Therefore, species avoiding the dry season respond similarly to dryness variations compared to those brevi-deciduous species that do not avoid the dry season. From this, our discussion said, shifts in growth patterns did not occur in monoculture treatments in response to dry season variations, but they did occur in mixed treatments, indicating that carbon allocation occurs in mixed treatments. Shifts in growth patterns in 5 species mixes indicated allocation to stem growth decreased in the dry and drier years, while respective growth of branches and roots stayed the same. Plots planted with 2 or 3 species mixtures, which are T plots, showed a similar decrease in the growth in both stems and roots in the driest year, and the branches remained largely unaffected. Therefore, the first hypothesis, which was that mixed species show stronger indications of carbon allocation in their changes in tree ring increment amongst organs, was in fact not rejected. Secondly, in our second hypothesis, Cedrela odorata and Luea siemanii had a similar dip in root growth and lift in branch growth in the dry season. This suggests that these species both had an increase and decrease in allocation of photosynthase towards roots and branches, respectively, in response to drought. Therefore, the hypothesis that these 2 species would show different responses to dry season anomalies was rejected. In conclusion, mixed species plantations may show adaptive responses to dry periods by shifts in carbon allocation patterns and therefore could perform better than monocultures in this regard, suggesting mixed species regimes can offer better use of limiting resources over monocultures. But more investigation is needed to determine if some indigenous tropical species are better adapted to drought than others. Thank you very much for your interest in this presentation. I hope that it was interesting for you.